Okay, so this was one of the slides that I spent a little bit of time yesterday, and just want to visit it again to give you some of the important steps. As you know, once an antigen gets recognized, it's processed in context of MSC class 1 or 2, and it's presented to T cell, B cell, and then these cells, lymphocytes, go through a process called activation of T cells. And this activation of T cell is the main process by which immune response or effector response will take place. Now, this is an oversimplified picture over here of intracellular events in CD4 T cell activation. And some of the steps have been omitted, but I would just want you to understand the first signal in this red box over here. You are familiar with that. MSC class 2 picks up a peptide, presents to a CD4 T cell. And then there is a second signal, which is from co-stimulatory molecules. And we also talked about the purpose of second signal is to enhance or to control the rate of progress of this event. And then we have intracytoplasmic chains. So the thing to understand in this case is that once there is a signal to these ITAM molecules and they get activated, they have an ability to bind phosphate. So you can see these semicircles, orange semicircles, they indicate phosphate groups. So once these phosphate groups are added, this leads to activation of molecules. And then there are kinases, for example, tyrosine kinases, especially SRC kinases, SRC, and two of them are important, FIN and LIC. These are important. They get activated and they start binding to those phosphorylated sites, as you can see. So once they get bound to those sites, and then there are some other enzymes like psych docking sites. The most important is ZAP70 because some of the patients may have immunodeficiency of this particular motif. So they will have problem of activation of cell. Now once the initial phase of activation of these tyrosine kinases or SRC kinases begins, so this activation will then bind to adapter proteins. So you can see these adapter proteins over here. So once they get activated, they will come up and bind to these adapter proteins. And the most important thing that ensues after that, then there are some MAP kinases, and you can see from some MAP kinases over here. So MAP kinases. So this is a sequence of events. What I want you to do is just to remember the sequence of events, and I'll explain that to you why that is important. Because once this activated adapter protein, they bind to those proteins like RAS and RAC, and you can see from RAS and RAC. This will initiate a cascade of events via activation of transcription factors, notably NF-kappa B and NF-AT and AP1, and that will then set up the order to the nucleus to start transcription of specific genes. So this is like, in a nutshell, very simple, oversimplified version of intracellular events. But at the same time, what you will see is that these adaptive proteins are not enzymes, but they basically contain multiple binding sites. So these adaptive proteins are not enzymes. They have a binding site, as you can see. And one of the most important things that they would do is that they will convert PIP2, which is phospholipid phosphatidylinositol, which is an important molecule, into diacylglycerol. So this is an important step. It will come in microbiology as well, and in the drugs as well. So PIP is converted to DAG, which is diacylglycerol, and then this will initiate two sets of events. And these sets of events are either activation of IP3 or PKC. So right now, once initial signaling pathway sets in, you have three different options for the cell signaling to take place. Number one, as you can see, you have MAP kinase cascade, and as the word suggests, cascade, and it's an oversimplified version. So this is not only one kinase enzyme. There are many different kinases, right? But eventually, regardless of what those enzymes are, they will lead to activation of transcription factors. So that's like a common end for both, for all the three pathways. Then you have an IP3 mechanism that comes from adaptor protein, and that comes from basically when adaptor protein bind to PLC and they catalyze the breakdown membrane phospholipid. So there's a phospholipid membrane, DAG, that is in the membrane, and these of the events are taking place on the membrane. So this is some kind of conformational change that's happening on the membrane. So that will lead to, number one, elevated calcium. So whenever we teach you drugs acting on calcium, or you must have heard of calcium channel blockers, or many different calcium sparing drugs. So many of the drugs that pertain to calcium, keep in mind that they are going through this pathway over here. The other important player in this pathway is calcineurin. So calcineurin, when it was discovered, that led to at least development of two drugs, cyclosporin A, very important, and tacrolimus. So these are the two drugs that were there, and they are used for prevention of graft rejection because those drugs bind to it. But eventually, they would also lead to transcription factors. Okay, and then finally, you have a DAG, PKC pathway. There are multiple proteins, and this would lead to, again, activation of transcription factors. So what you see in overall picture, which will come in B cell as well, that there is an initial signal, there is a secondary signal, they initiate protein tyrosine kinases, they initiate other sets of molecules, and that will allow phosphorylation of those intracytoplasmic tails of some of these molecules, and that would lead to activation of some other molecules, and eventually they would cause binding to adapter proteins, and then there is a sequence of chain of events that happen in the whole pathway all the way to the nucleus. Because even if I was to tell you that this T cell is going to proliferate, this happened through this pathway. If this T cell was going to show some receptor, it's going to go through this pathway. If this T cell wanted to help a B cell, it's going to go through. So all those immune responses that you normally see are coming from this pathway. This picture basically does not show you the B7-CD28 co-stimulatory molecule. So that's the second signal. So there is a B7 molecule and that binds to CD28. And this is another important thing. And one of the things that I'm going to talk about today is that once T cell get activated, it produces some protein we call cytokines. I talked about the IL-2, which is a growth factor. Okay? Now, next couple of slides, they are not posted to you. But again, keep that in mind. Just want to give you a perspective so that the drugs, where do they act? And some of the drugs which are there for cancer, for transplantation, for asthma, so on and so forth, they basically are coming from this pathway. Now, there are, I'm sorry, it's not that clear, but I'll explain that to you. So you can see again an antigen-presenting cell. It's presenting to a T cell receptor. And these are all different sequences of intracellular pathways. So what you are seeing is that there are 3 drugs which are acting over here. And there are some of the drugs which are acting over here. And some of the drugs are acting over. So the whole chain has those specific drugs. When we say signaling blocking molecules, and these are the drugs of the future. So all those drugs in treatment of AIDS, in treatment of many different diseases go through this signaling pathway. For example, if you want to give a tissue to a recipient, a donor, so once you give a tissue like a kidney transplant, so the recipient will reject it because it's foreign. So you want to suppress the immune system. So the main thing you want to do is that you basically suppress the intracellular signaling pathways. So these are some of the drugs that you may have. For example, since they go through different routes, so you have a choice of going to block one and the second and the third. So these are mostly, some of them are calcium dependent phosphatases, some block calcineurin, some block different molecules. And same happens in autoimmune diseases as well. Okay, now you can see from here that you don't have to remember, again, there was an oversimplified version, but basically all the drug companies right now, right, they are spending tons of money on that, exactly on the signaling pathway. So you can see story is the same. So you have a receptor over here, and they have 20 and 30 drugs lined to stop this initial action. And then each and every step that I just mentioned, where activation process takes place, they either want to stimulate it, if the need be, or they want to block it. And you can see all the way till the nucleus, because the reason is, I just want you to pay attention to this last one, because you want to modulate immune response. So if you get a cut on your finger and it's painful, it's inflamed, it may get infected, your immune system is going to take care of it. From that simple injury to a full-blown breast cancer, for example. So your immune system is going to fight it. There's nothing. So the drugs that you think that we're going to give it to them is to identify the deficiencies the immune system may have and to help them, right? So it may be, for example, the drugs may finally cause transcription factor activation. They may enhance cell survival. They may cause proliferation of the cells. And they may cause over here some of the angiogenesis. For example, angiogenesis is coming from those of you who are going to go in tissue injury, in dental school, for example. So what they do is when they take the tooth out and there's a gap over there, so they want to cover that gap. So you can have a tissue transplant, and sometimes they put a mesh over there. This mesh contains cells, cytokines, and growth factor to fill in that cavity. Or you have an injury somewhere, you want the repair to take place. So you want blood vessels to be formed over there. So you have a skin graft, and you want to induce proliferation of blood vessel, even after heart attack. So if there is a dead area, there are no vessels over there. The idea is that you want to create supply of blood via a process called angiogenesis. So that will also come from this drug. And then finally, you can see from here, it says, what does it say? Invasion. What does it say? Invasion metastases. So what's happening is in cancer. Because many a time, the cancer is located in the breast tissue. It may be very small, but it can metastasize into brain, into lungs, in different parts. So that also is one of the processes that we're looking at, especially for all the drugs that you see over here. So for at least this class, I would appreciate if you can at least remember the basic consequences. I don't want you to burden yourself with the name of drugs, but you should at least understand where is it coming from. One more example before I go further, again, just for your interest. Breast cancer. So you can see from here, when we talk of something for a targeted therapy, so we talk of a targeted therapy. That's another process that is coming up. And this slide is basically coming from the breast cancer. As you can see from here, we want to fight a breast cancer, so we have a choice even to block those receptors over here, or then go after those MAP kinases. We can even go for PI3 kinase, and even to such an extent that we can manipulate, and which I would not talk in detail because it's very complicated, that we can even go and change the sequence of event happening in the nucleus. So we can play around with the transcription of those genes with a PRP inhibitors. So this is the set of line of the drugs. Some of them are in use, and some of them are there in the progress to make sure that we can save those patients. So let's get back to the basics again. Many of the slides will be a little bit repetitive, and I will spend more time on the things that I think are new. So a resting cell gets an activation. It has a primary signal, it has a secondary signal, and signaling pathway sends the signal through whatever route we just discovered in the nucleus, and the nucleus says, okay, we want to produce a growth factor for T cell, which is IL-2. So this cannot happen unless there is a gene expression. Now, IL-2 is the most important growth factor. If there is no IL-2, there would not be activation of T cell. So this T cell, in a resting T cell, now will become an activated T cell. So what will happen is that now it will begin synthesis of IL-2, number one. It will also start synthesis of IL-2 receptor. Remember, normally, any given T cell, because some of these growth factors act on autocrine way. So they are produced by the same cell and act on the same, like an autocrine way. So you can see IL-2 receptor has beta and gamma chain, but this is non-functional. It would not functional unless you add a alpha chain over here, right? And this is good because you don't want your T cells to start proliferating without a good signal that is coming from intracellular pathways. So activation, first and second signal, and then synthesis of IL-2 receptor alpha, and this is now placed on exterior of the cell. And once IL-2 is released, it will go and act on the complete receptor to stimulate proliferation of T cells. Because what may happen is that you are unable to control an infection because your T cells are not proliferating. You are unable to take care of the problem if your T cells remain in a resting stage. You want the T cells to activate. Okay? And then you can see, then we can have a clonal expansion. We want T cells to expand, otherwise there would not be enough to take care of the problem that we may have. So once that cell becomes activated, one of the things that will happen, we call it effector response. So after the signal, these effects will take place in a T cell. And you can see number one will be IL-2 synthesis and proliferation. You saw that over here. There's going to be clonal expansion. Cell will divide and proliferate. And these cells can divide and differentiate to other effector cells that I'm going to talk about right now. And then again, these cells also will express those adhesion molecules which will guide them to move out of lymph node and go wherever they are needed. Okay, so this is a normal pathway where your T cell receive the first and second signal. Now also remember, as we discussed earlier as well, this is a repetitive slide, so it's going to go through that there are other ways to activate CD4 T cells as well. We talked about super antigens, and I said the most important is Staph aureus bacterial toxin. Sometimes these toxin activates, and then your cells respond in totally out of control way, and tons of those cytokines are made, and then you may go in shock. And some of the plant proteins, you come across plant proteins, they also can stimulate mitosis of these cells. So basically that's the mitosis of cell that's taking place, and we call polyclonal activators, and these are used in the labs. So we call it CON-A and PHA, which is phytohemagglutinin. So these are some of the plant proteins that we use. For example, if I want to test whether your cells can proliferate or not, so I'll take your T cell out, add these mitogens, these agents that are called mitosis, and see whether I'm able to get the proliferation or not. Then there are POC-weed mitogen. They are important because they can activate both T and B cell. And lastly, since all the T cell express CD3 molecules, so that is the basic of the, so they may be CD4 or CD8, but both of them will express CD3. So technically, if you bind to that CD3 molecule by an anti-CD3 antibody, so you will short circuit the signaling pathway, and then you can go from there. Okay? Now, let's see what are the major subsets that CD4 T cells after becoming active can do. Now remember, there are many different ways for us to categorize this CD4 T cell. We can either categorize based upon the surface expression of the marker. We can also categorize them based upon the cytokines that they secrete. And we can also categorize them by the effect of cytokines they take. But generally, these are two important categories that I want to divide it in. And this is important for you as a basis of understanding allergy. So we have, these are called helper cell. This other name for CD4 T cell is a helper cell. And these are first column CD4 T cells. So Th1, Th2. So helper 1, helper 2, simple classification. So what is the difference between helper 1 and helper 2? Well, helper 1 produces many different cytokines, but the signature cytokines that they produce are interferon gamma and IL-2, right? And we consider Th1 type cells as pro-inflammatory. So these are the cells that, because remember, if you have a cut and your immune response goes over there, so it is going to create inflammation, which is good. Because if there is no inflammation, the cell would never go over there and heal. But if inflammation stays for a longer time, you get infected, you get chronic inflammation, then it is bad. So you really have to have a balance between pro and anti-inflammation. So there are two different sets of cells and cytokines. One will cause pro-inflammation, the other will cause anti-inflammation. So you just want to make sure that there's a good sequence of events. So signature cytokines are gametron, IL-2, and these act on these cells. You can see they act. And the major effector response basically is that they mediate cell-mediated immunity, killing a virus or bacterially infected host cells. So you can see we need Th1 cells to boost our CMI, cell-mediated immunity, and fight against cancer and bacteria. So if you don't get that signal from the cell for T cells to proliferate, you will be unable to do that. And if you are unable to do that, you have immunodeficiency. Now, there are about more than 30, I would say, more than 30 different cytokines. I don't want you to remember all 30, but I think at least 10 or 12. I'm going to talk about that because they are used as drugs. Gamma interferon is used as a drug. IL-2 is used as a drug. Th2 type, they produce 4, 5, and 13. So you can come up with mnemonic to remember it. And they are very important in allergy, as you can see from here. So they are responses to worms as well and allergy. And they basically act on eosinophils, B cells, and then they will cause the B cells to produce IgE and IgG4. So you can see that IgE is an important antibody, and this antibody is produced when with the help of CD4 cell, and specifically CD4 Th2 type cell. Now, just imagine that if you want to upregulate this sequence of event, you have to stimulate these cells with these cytokines, isn't it? Right? But if you want to downregulate, you have to block these cytokines. So eventually, if you have a hypersensitivity response, your response is way above the normal. So what you would do is that you will come up with drugs that will block those cytokines. But if you have hypo, so you have a problem of autoimmunity and you have a problem that your immune response is not normal, so you have to give this response. So many times, for example, you already have a viral infection, your cells are fighting viruses, and you want to boost your viral immune response, you want to give gamma interferon, and this is given like IV. Now recently, I think, I don't exactly say 4 or 5 years ago, they came up with another cell, which is called helper 17, and they came up with 2 more cytokines, and they are important because they basically are, again, pro-inflammatory, especially to fungi. So you see from here that to a layman, it seems the same. So bacteria, viruses, fungi, parasite, they may look the same, but by the end of the day, they have a different immune response, and there are different cells or different cytokines that are taking care for us to fight these different microorganisms. And then there are a set of different drugs that we will come up with to fight them. And this was the most important cell that I did talk about earlier as well, Treg, right? So this is not a dinosaur, but it looks like a dinosaur. It really looks like Tregs. So they produce T regulatory cells, and they produce TGF-beta and IL-10. These 2 are very important cytokines. And I will talk about that in detail once I finish with today's lecture. And they are important. So what they do is that they basically regulate the function of other T cells. So they are like supervisor or masters or coach, whatever you want to say. You know, they will kind of regulate them. And you can see they mostly inhibit function of other cells, T and even non-T cells. There was a time we used to call them suppressor T cells, and then they disappeared and they came back now. If you are interested, it's not given over here. They are called CD4 positive, CD25 positive cells. So this is another marker, and there are many different markers that are important for them. Okay, so you can see from this slide, there is a T cell. It forms different subset. They have different cytokine. These cytokines, because cyto means cell, kine means action or kinetics, or something that will move those cells. So these are the cells they act on, and this is the major effector function that you normally see. Now, let's take, like, for example, T helper cells for now, CD4 T cell. And now in this case, what's happening is that the point is that this particular T cell, which was resting T cell, it is going to be differentiated into a different set of cell based upon influence of a cytokine. So this influence may be positive, right? And it could be negative, inhibition as well. So in this picture, you'll see if it's like a wavy line, it says inhibition. So once an antigen-presenting cell talks to a CD4 T cell, this antigen-presenting cell has a message for T cell to tell this T cell what is it, what type of subset that is needed, and what do we need to produce. Okay, so these subsets are produced in response to these sets. So you can see, for example, if you have IL-12, this will differentiate into TH1. Okay, and if you have IL-4, it will differentiate into TH2. If you put TGF-beta and IL-21, it's going to become TH17. And then TGF-beta is required for T regulatory cells. And then these subsets, so this is how it goes. So the cytokines act on T cells, and cytokines are produced by T cells as well. Dendritic cell or antigen-presenting cell make those cytokines, and so it's like both autocrine and paracrine way that these cytokines act. So they are very important in today's modern therapy. So we are using these cytokines for treatment of many different things right now. So it's good for us to understand actually what is going on and how this body deals with it. Now you can see from here, once you form TH1 cell, this is known to produce gamma interferon, we already know that. And this will have a inhibition on TH2 cell. So TH1 cell and TH2 cell, they produce something. So they themselves are pro-inflammatory, but they may have an opposite effect on each other. So in this case, you can see that TH1, which is pro-inflammatory, is going to inhibit TH2. On the spin side, TH2 is going to reverse TH1. So they kind of go totally opposite. Okay, and some of the effects are still under research. As you can see, IL-17 is one. TGF-beta is important because remember that T regulatory T cells are controllers of T cell. So it will control all of them. So all of them basically, if they go out of control or produce in excess, so they will have a negative effect on all of them, TH1, TH2, TH7. So all these three are regulated by T regulatory cell. And this is an important concept for you to understand, especially before we start teaching you immunology of cancer. You would not understand why cancer is causing problem and what if you don't understand the role of T regulatory cells, because that is going to be the trendsetter for you to understand the cancer therapy. Now, what I've shown you so far, that CD4 T cell is activated, it produces cytokine, it has subsets, and then again, some of the functions are there which were not evident in the previous figures, as the word suggests, helper. So this is a helper cell. So you can say interaction of CD4 T cell, so they help a B cell. And one of the things that if you remember, I said that though B cells make antibodies. So what do you call a B cell that is ready to synthesize antibodies? What is the name of that cell? Plasma cell. Very good. Plasma cell. So plasma cell. So remember, B cell is going to expand and start making those antibodies. And this is under the influence of CD4 T cell. Now a difficult question. So what type of antigens do you think are going to be dealt with in terms of a response when we talk of CD4 T cell? So I'll just give you a clue. Do you think that it is a thymus-dependent antigen or thymus-independent antigens that these T cells deal with?
Thymus-independent.
Opposite. Dependent. So this thing, keep in your mind, because they usually go for the protein antigens, right? And protein antigens are the one that are important. Otherwise, all these things will be taken up by other cells. Okay. Now that's, again, you missed the point. I asked the question and answer is the next line. You just have to kind of go a little bit ahead of me, right? So this kind of cooperation which is required for the B cells, for T cell, especially when they are thymus-dependent antigen. The reason being, can somebody tell me the reason why the B cell would need a T cell help in responding to thymus? Yeah.
Because T cells mature in
Thymus, well, both of them matured. But there's another reason, yeah.
Because T cell helps B cell to make antibodies.
That is correct, but why? Question is why? Say it again? It has to be specific, I agree, but what actually triggers? Why do we need a T cell help for B cell, especially when it comes to a response of the, yeah, somebody raised his hand.
Immature B cell?
Could it be the ligand of MHC?
Could it be? That's like you're not sure, right? And you say, could it be? Well, the reason is, if you remember, the analogy I gave to you, that B cell is happy to take anything in, but T cell just wants protein. So if the B cell is going to react and respond to a protein, so B cell needs T cell help. Because B cell is good for carbohydrate, lipids, and other things. But whenever it comes to proteins, right, because proteins have to be presented in context with MSC class 1 or class 2. So that is not the job of a B cell. B cell can work independently of that. It's only T cell that wants to see a protein in context with MSC class 1, even to produce an antibody. And then again, well, it actually shows over here as well. But let me give you an example. Does that make sense? So everything kind of relates. So the way your chapters are designed, one thing will add. It's a cumulative effect that you will have. So if you understand one concept, it will be easy for you to understand the other concept. Okay, probably when a T cell needs help from a B cell to cooperate in antibody synthesis, so we call it a linked recognition. So I just said it needs help, but you need to know what exactly is going on in terms of physical term. I just told you one thing as a rule. Whenever B cell needs to produce antibodies in response to protein, it needs T cell help. So far, so good? Now, example. So this is a protein, and you can see this is protein, has different epitopes, surface markers. And also remember the size of amino acid in terms of epitope that these express. It has to be small. So antibodies or cells will just go and bind their receptors to that dock. They'll just see that and just bind to that, right? So in this case, what's happening is that once this protein has been processed by APC, now it presents two different epitopes, epitope A and epitope C. Now epitope A is specific for an epitope which is there on the T cell. At the same time, B cell will look at the epitope C because the receptor for B cell is already there. Now, what is happening is that they are getting two bits of information from the same protein. And they are seeing two different epitopes. right? No, they have to make a move in terms of immune response, so they need to cooperate. So this is what happens. You have a T helper cell, B cell cooperation, especially for synthesis of an antibody, because you want to make a very specific antibody. Okay? And the other important thing is that, let me show you in a better picture, maybe it will make sense to you. Now you can see from here, a B cell captures an antigen via antigen-specific receptor Ig and internalizes it, right? You can see it is internalized, and then the antigen is broken up to small chunks of peptide in acid vesicle, and then basically it is associated with class 2 and comes be expressed. So why do you think that a B cell is able to present a particular peptide in context with class 2? Why do you think? Why did not B cell go for class 1? Why did it choose to present this particular antigen in context of class 2? No, I said B cell. Why did B cell decide to process this antigen and present this in context with class MSC class 2? Louder and raise your hand so I know who's the one that should be credited. Maybe I'll give like 1 extra credit point to you if you answer me. Yeah. Because I told you earlier, yeah. Okay, let me give you, don't say think green, think simpler. One of the information in concept forming was exogenous antigens are presented in context with MSC class 2. That's what we decided earlier, right? Since this is an exogenous content, and it is being processed in an acid vesicle, it is going to be presented in MSC class 2. Okay? All right. Now, it is presented to a T cell, same story. You will see over here is a CD4 T cell, and it's talking to B cell. So these cells talk to each other. In this case, this is talking to a B cell, this is a protein, and then it is broken down, presented to in contact MSC class 2, and then the rest of the docking site are pretty much the same. The only new thing that is added to this figure is there is a cytokine receptor on the B cell, and there is a cytokine secreted by the T cell. So there is a cytokine basically important, because remember, what is the normal antibodies that the B cells produce? These are the questions I'm going to ask you. Whatever question I'm asking you. What are normal antibodies that the B cells produce? Very good, IgM and IgD, because they get expressed on the surface of that. And now you want them to switch to another class of antibody like IgG, E, or A. This is called class switching. Now, how would that happen? This will happen, for example, by this mechanism. A cytokine will be secreted by the T cell, and a corresponding receptor will be expressed. So why this receptor is hatched? Why this receptor is hatched? Because this receptor is not expressed on a unactivated or resting B cell, because there is no need for that receptor. So once the T cell engages, primary signal, secondary signal, now this is the time for the B cell to express that signal, because if there is no cytokine receptor, this cytokine will have no way to act. Okay? Now, these cytokines then lead into which particular antibody needs to be produced. For example, you have a problem with IgE, you're not producing IgE. I want your system to help you out so you can take control of your allergies. I want a device with a drug for the allergy. So what I would do is that just knowing cytokine is not enough because there are tons of cytokines. So I will go and give you IL-4. So once an IL-4, if this happens to be IL-4 specific receptor, this happens to be IL-4, this will then start producing IgE or IgG4. And if it is a gamma interferon, so you can see that a particular antigen cytokine has a particular specific effect. So this is where the drugs and the cytokines are coming from. Okay. Now, let's give you another example of this time CD8 cell. What was the other name for CD8 cells? Cytotoxic. Very good. Cytotoxic or killer. Better cytotoxic because they are going to kill the cells, right? As compared to helper. Now, so you can see from here there are three different mechanisms where CD8 cells are talking to the cells. Number A, there is a virus or virus-derived protein, and dendritic cell will take it in, present it into class 1, because you know viruses are dealt with class 1, and now CD8 cell comes and look at it class 1, because CD4 doesn't care. CD4 just sees class 2. And CD8 will come, it will go through the same activation processes I just mentioned, intracellular pathway, and then it will start producing IL-2 receptor, and then we will have a CD8 effector response. This is one mechanism. The other pathway is that the virus is taken up by an immature dendritic cell, and then, if you remember, immature can present in context with MSC class 1 and class 2. When it decides to present to class 2, of course, CD8 cell will come and dock in. Otherwise, CD8 is enough to take care of that. Now, the question is that in this figure, we figured out one of the processes that happen in effector response is that IL-2 receptor is being expressed, which is a growth factor. Now, this IL-2 receptor would need IL-2 to start proliferation, but this IL-2 is coming from CD4 T cell. Okay, and finally, if there is, for example, in this case, a virus-infected target. So this target is virus-infected, and then there are these receptors expressed. One of the receptor, of course, will be a receptor MSC class 1, you can see from here. But viruses, basically, when they infect the cell, and we'll discuss that in detail, they present special kind of receptors on top of infected cells. Just for brevity's sake, one of the things that they do is that they express a perforin pore. So they open up a pore into the cell. The other thing that they express is called Fas ligand. Fas ligand. Now, when a CD8 cell docks with a virus-infected cell, right, it is going to, of course, take the first signal and second signal. And CD8 cell, because it's a cytotoxic cell, it contains granules, and these granules will be channeled through that pore into the cell to kill the cell. That's why you got herpes labialis, you get this sore, cold sore you call, this other tissue damage because your virus is chewing up your cell. And this is what is happening because your CD8 cells are coming and they are attaching to that and they are killing your own cell. Okay, so this is a typical story that normally you see in terms of virus-infected cell. Okay, one last slide, if you just give me one more minute, and I'll be done with that, because this is something that I already discussed with you, nothing new. A conjugate vaccine. So these are the vaccine. Remember, if it's a simple vaccine, you have a polysaccharide, you have a vaccine that will only stimulate B cell. But we would rather have a conjugate vaccine where we attach a carrier protein to it, because proteins also make good vaccine, because then we'll initiate T cell responses. That's the whole upshot for that. So you will have a more robust kind of a thing in terms of response. Okay? And this is basically pretty much the same. The only difference is instead of T cell, it says B cell. That pretty much the same. And this slide I also discussed earlier, where I said if you express a co-stimulator, you would enhance the effect by complement pathway, right? The slide I did not present last time, that this is a stimulatory pathway, but it could be an inhibitory pathway as well. And that happens, remember, when they are phosphatases. And there are some of the other receptors which are there that will inhibit immunoglobulin synthesis. So we have a dual mechanism because we want to control everything. In this case, we can upregulate, stimulate when your immune system is not doing what it's supposed to do, or when it is going out of control, we can put brakes. But we have all different drugs. They are acting in each and every step that we have in the intracellular signaling pathway. Thank you.